that first you have expression of the heavy chain, and then you then get expression of the kappa light chain, and it says review this kappa light chain. If that is a successful, creates a successful pairing and doesn't recognize self, then you would go on, and that would be that would go on, and you wouldn't continue on with the process. But if it was a non-productive allele, then it would go on, and it would choose possibly another light chain. So you re-enter in the process of you would have BDJ recombination happen again, and choose different pairs, and then you would, and if that fails, then you would go on to lambda. And so it's pretty tightly controlled so that you only get expression of one or the other, and that expression doesn't recognize self. And so, again, you can have recombination occur initially, and that first rearrangement between, in this case, B3 and J3 is productive, but combination results in an autoimmune antibody. And so, like we discussed with T-cell development, you have some negative feedback that then throws the cell back into BDJ recombination, and you can get 2 recombining with 4 in this case, and then that would, if that is a successful pairing, it would go on. If not, it would go on to do the same thing with your lambda, light chain, and so on. And so this is kind of this, throwing it back into the recombination machinery is kind of this last ditch effort to salvage the rearrangement. Okay, so how is this tightly regulated? You don't have to memorize this, but you do need to know that there's promoters involved, enhancers, and then locus control, basically where these things are located. So before recombination, there's different promoters in front of these different B's and D's and J's, and they are too far to work together in conjunction to initiate transcription, but when they do move closer together between the promoters and the enhancers, and moving those locations closer, that's where you're going to start getting transcription. And then if that transcription is successful and you have an allele that does not that is successful, it doesn't recognize self, then it'll go on. But if not, then it'll start the VDJ recombination again. And it's a pretty complex process, but all you need to know is that it's tightly controlled, and how it controls that is based on location of these promoters and enhancers.
Do we need to know what kind of promoter and enhancers are?
No.
It's just
Yeah. So a promoter is just, and I'm pretty sure you probably learned this in a lot of your other classes. A promoter is going to recognize certain sequences that are on the, and then it's going to recognize those sequences and bind, so that promoter is going to bind on that. But then it also requires other enhancers that are going to bind to those other transcription factors and facilitate bringing all the other transcriptional machinery into the mix and initiating transcription. So there are promoters and enhancers all throughout, and you can see that this is just showing you locations of different promoter regions, enhancer regions. There are promoter regions in front of the constant regions, in front of both the V, D, and constant regions. And by bringing those closer together and close together to the enhancers, you're able to get expression of that protein.
So you need a one V, and you have min from your method. How do they combine?
You need them all in order to get expression. So they bring together the machinery to start transcription. So as long as they're separated, so that's why if you get recombination of this guy with either one, let's just say this guy, then you're bringing these two promoter elements closer together.
One for each, that's what I,
Yeah.
one for each.
And then recombination with then the J will then now bring it closer to this enhancer region, and you can now get expression of that. And that whole thing will now be closer to these constant region promoters, and you'll get transcription of those. So you have them for the light chain as well. Both these light chains have the same type of situation where you have promoters that will be brought in closer to enhancers, which will then initiate transcription. And you don't need to memorize the location of these. Okay, so heavy chain, let's see. So then the next part is now we have different constant regions that are going to dictate what type of IG molecule you have, IgM and IgD. And so now we have a second kind of, you already have your TC or your, your Tcell receptor, and it's specific for a specific antigen, and that's not going to change, but then your class of your antibody is going to be dictated based on mRNA splicing. So now this is at the mRNA level. You have your heavy chain BDJ is put together, but the spacer RNA sequence between that BDJ and the C region exists, and then that splicing removes that spacer, leaving the BDJ and then your constant, and then your mRNA is ready to be translated, and that primary transcripts for IgM heavy chain may result from transcribing through both the IgM and IgD constant regions. So depending on which C segment becomes polyadenyl, so you get polyadenylation, either the IgM or IgD, and you could end up with either IgM or IgD based on that. So we're going to go into class switching later on, but just know that that class switching or choosing of whether you have IgM, IgD, this is happening at the mRNA splicing level.
So when you say switch class switching, that's only from IgM or IgD?
No, yeah.
And fine.
Okay, so mature B cells can express both IgM and IgD antibodies, and then this mRNA splicing based on polyadenylation will occur, and you can get one or the other. It's just an example of that. And then also that same type of post-transcriptional processing is going to be involved in whether mRNA splices the mechanism that allows these IGs to be membrane bound or secreted. So again, they're going to include the portion. In some cases, they're going to include that portion that has that membrane region that is going to be that hydrophilic or hydrophobic region that's going to be in the membrane, or they're not going to include it. And so based on whether you get that polyadenylation and keep that portion is whether, so again, the decision to secrete an antibody or to keep it membrane bound is based on that.
How do they decide to have membrane bound and it's like a 4D?
There are other proteins involved in polyadenylating that mRNA, and so that polyadenylation is based on post-transcriptional regulation.
But if they have polyadenylation, that means they are membrane bound?
Yep. So if that region is polyadenylated, it's going to go ahead and
They have a few amino acid in the
Yes.
cytoplasm.
So T cells, So that's B cells. T cells are going to be very similar in how they rearrange or rearrange and recombine their gene segments in order to make that T cell receptor, in order to get that variety. And when they were, so B cell receptors, they kind of discovered those first, but then as they were looking for T cell receptors to see how those were regulated, they used monoclonal antibodies bound to T cell receptors to purify them, and then once they purified them, they could study what they looked like. They found to be, and so they found out that these were also Ig-like molecules. They had an alpha and a beta chain. So similar to the light chain, so the alpha chain was similar to your B cell receptor light chain. The beta chain of these T cell receptors were similar to the antibodies heavy chain. And the beta chain gene setup was discovered first. And then while they were looking for alpha chains, they actually found gamma. So you might remember that the majority of our T cell receptors are going to be alpha beta, but there are also gamma delta T cell receptors. Those don't have quite the same variety as the alpha betas, and play a different role. We don't have as many of them, but that just happens to be the order they kind of discovered those in. I'm not going to hold you responsible for that. So T cell receptor genes undergo rearrangement similar to those Ig genes. So we've got the arrangement of the T cell receptor genes, and in the last diagram was very similar to what you saw with the Ig genes. And so you have on humans, you have an alpha chain on chromosome 14, and the beta chains on chromosome 7, the gamma chains on 7, and the delta chains on 14. And then your betas are like your heavy chains. You've got BDJ recombination. You have 40 to 42 different functional variable regions. You have 2 functional D regions, 6 J regions. So very, very similar to what we saw with IGs. And then you have the lighter, the alpha, which is more similar to the light chain of IG, where you have 43 potential Vs and 50 Js. So a lot of variety in these, which might lead you to suggest that you have more diversity in T cell receptors than you may have in IGs, but because of and we'll discuss this, you have somatic hypermutation, which also occurs in B cells after they encounter antigen, and that gives them an increase in potential outcomes, whereas with T cell receptors, you don't have T cell receptors undergoing somatic hypermutation. So that's a little different for T cell receptors, so maybe that's why we've developed to have such a variety, a little bit more diversity in those T cell receptors. Yeah?
So what do we need to know about this kid?
You need to know that the betas are similar to heavy chains and that you get BDJ recombination. You need to know that, in general, T cell receptors have a little bit more diversity. They have 40 to 42 different Vs. I don't know that I require you to memorize these numbers. It's kind of good to know that you have 2 potential Ds, and 6 Js is a pretty easy number to remember. And let's see, what else should we know?
So
as you can see here, also the alpha and betas, you can see that there's a lot of diversity, but then your gamma deltas are your gamma deltas don't have that same diversity. They don't play the same type of role. They almost resemble more members of an innate system where they are able to recognize specific elements of pathogens, but they don't have a lot of diversity. So they're almost, in a way, even though they're not, they're in a way more resemble your innate immune response as opposed to this adaptive immune response. So T-cell receptor genes are located somewhat differently between mice and humans, of course, just like they were with Igs, and you don't have to memorize the differences between mouse and human. We're going to focus on human. Their T-cell receptor genes, they undergo the same type of process of rearrangement. You have your RSS locations allow for these recombinations. So, for example, you would have recombination of B with D or J, but you could also get recombination. So here you have your alpha similar to your light chain, where you get recombination with B's and J's. Here they're showing the beta can recombine with B and D, and then the D's and J's can recombine. Same with the gamma, but then the delta, here you can see because of the arrangement here, you have the potential that where if you have two D's, those D's could actually, you could get two D's recombining for both the beta and the gamma, and you do see that with gamma. So you could get potentially one or two D's in that mix with the delta, and we do see that with beta. It's actually possible based on sequence, but it's you don't ever see that. So looking at the process of rearrangement and kind of comparing, do B cells, B cells have EDJ genes? Yes. T cells do as well. Light chain segments, you've got the kappa and the lambda variable regions encoded by D and J segments, and then you have alpha and gamma variable regions encoding D and J. And T cells, heavy chains, B heavy regions encoded by BD and J segments, whereas in T cells it's the beta and delta that will recombine. They are both dependent on RAG1 and RAG2 for these recombinations. So a RAG knockout is going to not only be devoid of B cells, but also T cells. So when you're talking about RAG knockout mice, that people do studies, a lot of the initial immunological studies where we were studying adaptive versus innate immune system use these RAG knockout mice, and therefore you would have mice that had an innate immune system but did not have this adaptive immune system because they didn't have B or T cells, because these rearrangements never occurred.
Rag knockout mice is like similar, resemble to the innate immune response.
They have an innate immune response because the RAG gene is only needed for the recombination of those T cell and B cell receptors.
So that means
they have NK cells, they have all the innate immune cells, macrophages, they have granulocytes, so they have all the populations of cells, but they don't have B cells and T cells that are functional.
So rag knockout mice, we put a experiment, is just like you are studying, you put even a VDJ there and to see how you just doing like adaptive.
Yeah, you're just looking at adaptive. They do also use them for studies where you're looking to study a specific B cell or T cell, because you can put that in there knowing that the only response you're going to see is from a specific, whatever you put in there, because they don't have their own population. So a lot of people
It's
use
a model system.
Yeah. So we have these also, remember we talked about junctional diversity, where you add nucleotides onto the P or the N addition. So you see that in both B cells and T cells as well. And then multiple D regions per recombined chain. You don't see that in IG, but you can see that in delta T cell receptors. It's possible in beta chains, but we don't see it. So these are kind of just, this is a good chart to kind of know the differences between B cell and T cell receptors. So same type of allelic exclusion of receptors is absolute B cells. So you will not see one heavy chain with two different light chains expressed in one cell. Each cell is going to have just one heavy chain, just one light chain. Whereas with T cells, allelic exclusion of that alpha, which is our light chain, is it's not absolute. So you can get some, you can have two different light chains or alphas expressed for T cells. This is not seen for B cells, but for T cells you can see that. B cell receptors, another difference is that you can have membrane-bound B cell receptors, but you can also have secreted B cell receptors. This is something we don't see at all in T cells. So your T cell receptors are never going to be secreted. They're always going to be in a T cell, not floating around. Constant reagents determine, nature of the constant reagent determines the function. Again, B cells, remember we have different constant regions, which can make IgG, IgM, IgD, all these different IGs. For T cells, you only have one constant region, and it basically expresses, it's basically the same in all T cell receptors. It's going to have that intermembrane region that's going to help localize your T cell receptor with that T cell receptor machinery that allows for when they see antigen, they're going to then, again, remember the T cell receptor cascade, expression cascade, they're going to associate with those other molecules, and you're going to get phosphorylation events, and then you're going to get downstream events that can basically upregulate promoters that will now tell the cells to proliferate, tell them to secrete cytokines, and all those other things.
So, when you say T cell receptor, that means they always come with a CD3?
So the CD3 is a separate gene, but CD3 will associate with that T cell receptor. So their constant region is always going to have those binding sites for CD3, so that it will associate with that.
So the TCR always a membrane part?
Yeah. And then here's the other big difference between B cell receptors. T cell receptors is B cell receptors undergo somatic hypermutation following antigen stimulation. So you have all this BDJ recombination, different translation of events that will choose your whether your IgM or IgD. But then once those B cells come in contact with an antigen, they're going to go through another process where they actually induce mutation events within that TCR or within that BCR, and they will then, now you have a further selection event. So those that still continue to bind to antigen and with a higher specificity will go on and proliferate, and those that don't successfully mutate to have better binding will not. Whereas T cell receptors, you get what you get, and they don't undergo that type of a process. So you can actually have much more diversity in your antibody receptors, but initially it looks like you probably can get a little bit more diversity with your T cell receptors.
So which one has more diversity between these?
Well, B cells end up with more diversity because of somatic hypermutation. So same machinery doesn't recombine Ig genes. So they have different machinery, but they also have the same type of process, and you don't need to know which genes are different or whatnot. T cell receptor gene expression is also very tightly regulated. So again, similar to what we discussed with B cells.
Is the difference in the T cells and the B cells? The reason why the B cell has more function because it begins in the bone marrow and then moves from the bone marrow to the thymus to evolve further, and that reduces its activity or its mechanism.
No, it's just different. Where the two are developing, like you said, B cells are developing in the bone marrow and going to the lymph nodes. If they see antigen there, they're going to undergo somatic hypermutation. difference they just have different roles, different functions.
So regardless of its originating terminal, functions discerned by its developmental process or design.
So
Its mechanism of regrowth. Is it the mechanism of regrowth, regeneration, or is it the actual progenitor cells that cause the selectivity?
So are you asking where the selection occurs?
Right, but why the selection differs its mechanism?
Oh,
Or differentiates its
different.
mechanism?
So why do they have? I suppose they have different mechanisms because you don't want to have. So these are both lymphocytes. You don't want to have a B-cell receptor and a T-cell receptor, and so they kind of have these tightly regulated mechanisms to where they're going to make a decision whether they're going to be a B-cell or T-cell pretty early on. If they, and that's probably why they don't share the same transcriptional regulators, but it also could be because they've developed on different chromosomes, and over time that has changed, but it probably does have some functional reason. But that also, they are going to be developing the bone marrow versus thymus, and that probably also, there's probably a reasoning behind that as well, because in the thymus you're set up certain cell infrastructure to have this positive and negative selection occurring, whereas in the bone marrow you're more geared towards selecting B cells that don't have auto responsive. So it's the machinery, again, because they're tightly regulated, they do have different transcriptional factors, but whether, yeah, I mean, I don't know if that answers your question or not.
Kind of. I mean, I know that in the text it goes through a little bit more about like the thymocytes and bone marrow sites and how the different functions of growth there can determine. But I know that the T cells originate in the bone marrow, so I was wondering why T cells get a little bit less active as they mature in the thymus. You figure that with more development, there's more control over whatever the situation is. But it's the exact opposite. B cells have a little bit more control because they have those secretory and membrane bound.
Well, we're going to talk about why that is. And having both of those kind of allows B cells to play a role in different responses to exogenous antigen or cells that have been infected, whereas T cells really target cells that intracellular. So T cells are very much just focused on cellular targeting cells, whereas B cells have both the function to target cells, but also to target proteins, to target other pathogens that aren't intracellular pathogens, to target toxins, to target, it's just, we evolved to have these kind of two different systems. They work together, they can communicate together, but they kind of have different roles. Yeah. So we're going to go into that a little bit more when we talk about T-cell antigen processing and presentation. They developed separately because, and they have different functions altogether. So B cells are not necessarily dependent on an MHC molecule presenting antigen. B cells can recognize antigen in the context of almost anything. So that's another difference.
So they don't depend on MHC?
The B cells don't, they can recognize whole proteins, so they can recognize proteins that aren't attached to a cell, that are maybe a complex of complement. They can recognize. So B cells, once they have that antibody, they can recognize a pretty huge range of things. Doesn't have to be a cell, whereas T cells are only going to recognize antigens that are presented to them. And that's what we're talking about next, is antigen processing and presentation. So antibodies have this, let's just say this region that's going to recognize antigen. That could be a soluble antigen. And so, and that's kind of why you can have a soluble antigen, or you can have an antigen that's attached to a cell. So these could play either a role in eliminating soluble exogenous antigen, or they can also phagocytose cells that are cell-associated antigens. So these could be antigens that are coding a cell. So antibodies you can have a B cell attached to this, or this could be soluble. So for example, soluble allows for the diversity of me having a soluble antibody bind to this cell, and then I have other cells that will recognize that heavy region, that constant region bound to a cell, and will phagocytose that cell. So it could be a B cell that phagocytoses that cell. It could be a dendritic cell that phagocytoses that cell because there's an antibody on it. So B cells, they can have kind of this cytotoxic or phagocytic effect, but they can also mediate an immune response against cells or against antigens. They can go around collecting antigens and kind of soaking them up, which is what you want when you have an anti-venom. You know, you want to be able to kind of not have that protein doing damage to to cells, and so antibodies really have a lot of different functions. Whereas T-cell receptors, they stay bound to a T cell, and it gives you a little bit more control over, in the long run, about whether you make a decision to kill a cell or not. Whereas the antibodies, they're just seeing antigens in whatever context they are. So let's go ahead and jump into T-cell receptor or antigen processing and presentation. So we're going to talk about MHC molecules, major histocompatibility complex, and antigen presentation. So we've talked about T-cell and B-cell receptors, and now we're going to talk about the other end of the T-cell receptor, the MHC molecule. And we have at least two major MHC molecules we're going to talk about. Our MHC class 1 molecules and MHC class 2 molecules. In general, it's good to remember kind of what I drew up here. All of the cells of the body express MHC class 1. So that's kind of their way of being able to alert the immune system as to what's going on in the cell. They're going to have, those MHCs are going to present antigens out on the cell surface, and then T cells can come and kind of see those antigens. If they're self-antigens, we shouldn't have T cells that recognize self-antigens, so you're not going to get killing of those cells. But if those happen to be viral antigens or antigens that are expressed because this cell is cancerous or is a tumor and is expressing certain proteins or genes that it wouldn't normally express, then that's going to kind of clue in T cells that something's going on in that cell, and they'll be able to recognize it, target it, and eliminate it. So that's happening all the time in your body. So that's MHC class 1. How we kind of also, this second line of defense, or the second way of the checks and balances, you have these helper T cells that are needed in order to really induce a cytotoxic response. And these guys will only see the MHC class 2 molecule molecules, and those MHC class 2 molecules are primarily expressed on antigen-presenting cells. And so the antigen-presenting cell now is this mediary that is going to, its job is to take up exogenous antigens and then present them again via MHC class 2, and we'll talk about that process and how that happens. Whereas MHC class 1 their job is to present endogenous antigens, what's going on inside the cell, whereas these are looking at what's being brought into the cell. And that gives a little bit more control. These APCs will pick up, they're brought in when there's danger signals involved. They're picking up antigen from cells that have apoptosed. So they're these guys are a little bit more alert to things that are damaging. Maybe tumor cells, for example, start going out of control, start pushing on epithelial cells in the area, start to cause inflammation, start to kill other cells. Dendritic cells would come in, be brought into that area because of the inflammation that's occurring, because of apoptosis that's occurring, because of cell death, and they would start picking up antigen, present it, and then those CD4s would become activated. They would start secreting cytokines that would tell the CD8s in the region, go ahead and whenever you see that, go ahead and attack that cell. Whereas if CD8s are just going around looking at cells, they might say, hey, I see this antigen, but there's nothing bad's happening, so maybe I won't do anything about it. So we have this pretty tight regulation. So we have MHC class 1 and class 2, and again, just in general, MHC class 1 is going to be on all cells, MHC class 2 only on antigen-presenting cells, and MHC class 1 is involved in presenting endogenous antigen, whereas MHC class 2 is involved in presenting exogenous antigen. And that doesn't mean, and this is actually, so basically that's a whole lecture, you guys won't have to listen to it. But we're going to go into a little more detail of how it happens. Also, you should know that and I'm just saying this because this became an issue with my thesis, is that when you talk about MHC, we talk about MHC class 1 and we teach it and we say MHC class 1 presents endogenous antigen and MHC class 2 presents exogenous antigen. When we teach that, that's what we teach, but that does not mean that these are absolutes. MHC1 can present exogenous antigen, but more to the point, MHC class 2 can present endogenous antigen, and oftentimes it will. And in some of those cases, that can actually lead to autoimmune disease, or in other cases, it can alert cells to potential hazards that are occurring inside an APC. But when we talk about any of these processes, we kind of look at them like on-off, black and white, and sometimes they're a little bit leaky. So just letting people know that. So class 1, let's talk about class 1. These are also, like we saw, these are also members of the Ig superfamily. Again, these are not T-cell receptors or antibodies, but just members of this Ig superfamily, possess Ig domains. There's a larger alpha chain. Now these, again, don't think of these as having diversity because we don't have T-cell receptors or B-cell receptors on all of our cells. These are, the role of MHC molecules is to present a wide array, not to have specificity, to really have the opposite of specificity, to have diversity in what they present. So you've got 3 external domains within that alpha chain. They're approximately aligned. You don't have to memorize this. You have a transmembrane domain, approximately 25 amino acids, cytoplasmic anchor section of 30 amino acids, and then these are all play a role in the function. So these are again, these are not secreted. These are going to be in membranes. It's gonna allow them to traffic, and when we talk about antigen processing and presentation, these are made, they enter the ER, the endoplasmic reticulum. They're going to traffic from the ER up into the cell surface, and so they need to have certain elements that help those to get trafficked and processed efficiently and get to the right place. Also, these can, just like T-cell receptors can associate with lipid rafts, MHC molecules can associate with lipid rafts, and that helps to localize more and more together in a synapse with a T-cell receptor, with a T-cell and an antigen-presenting cell or an epithelial cell or whatever. So the alpha-1 and alpha-2 domains form a cleft region that binds 8 to 10. So this you do need to memorize 8 to 10 amino acid long peptide. So here's your antigen. So the big difference between MHC class 1, so MHC class 1 is going to have this basically kind of this structure where you have this alpha and then you have this beta, 2 microgalactose. So you have an alpha and a beta, but really the alpha is going to play the major role in this antigen binding region. And it's not going to bind antigen like an antibody does, where it's specific. It's going to have these little anchor residues that are going to be specific for certain amino acids, but then all the rest, and notice that I'm drawing this with little walls up here, and this is how you can make sure that you only have little peptides of 8 to 10. So what you get is, and we'll talk about that, is everything that's in the cell, proteins that are being made in the cell or recycled. We're always making new proteins. We're targeting old proteins with ubiquitins. Those go into the proteasome, get chopped up into little pieces. Now it's these little pieces that are going to get loaded into our MHC molecules and then delivered to the cell surface, basically just saying, hey, this is what I've been making. This is what I've been up to. And so these little peptides, they may have the same 2 amino acids on either end, or same couple of amino acids that have similar properties on either end, but all the amino acids here in the middle are going to vary. Another thing to remember is that we have a lot of alleles, different alleles. This is this is where our diversity comes in. This is why that you have to have donors matched for organs because of these MHC molecules, because we all have different alleles of these MHC molecules, and they're going to present different peptides based on these sequences here, based on what their specificity is. Even though it's very loose specificity, you're presenting different antigens than I'm presenting, and therefore that's an advantageous genetically speaking or evolutionarily speaking, because if the Black Plague comes along and I don't happen to present an antigen that's present in the Black Plague, I'm going to die from it. But you might present an antigen that is being expressed on the Black Plague, and so you survive it, and your progeny will now be better equipped to deal with that, and humans continue to exist because we have this diversity of what antigens we can present. And and that's where a lot of, like in Europe, actually they blame the Black Plague on Europeans having a pretty narrow range of MHC specificity compared to maybe other places, because people who were able to recognize antigens on the Black Plague, they survived, and their progeny continued to have the same kind of alleles, and so that kind of dictated what alleles were prevalent in Europe. So that's just a side note, I guess. Let's look at the picture of this. So you get this class 1 molecule that has these alpha helixes that kind of create this little. So you've got this nice little area for an antigen to sit in here, and it can only be so long, 8 to 10 amino acids long, and it's going to have some specificity for maybe something here in this beta sheet, maybe in this alpha helix, and then all the rest is going to kind of stick out and be presented to T-cell receptors that may or may not recognize that. And here again, you have all of this, all these alpha 3, alpha 2, alpha 1, and this is all in your alpha. So your alpha makes up that entire groove in the MHC molecule, whereas you have this beta 2 the beta, so kind of just sitting there kind of stabilizing that groove. And so there is an alpha and a beta, but again, for MHC class 1, it's this alpha that is making up, is where all this diversity and specificity for antigens is going to be.
So the alpha 1, 2, 3, are they similar?
These are just domains within the alpha protein. These are,
So one protein is
yeah, one protein. This is all one big protein, just showing you different domains within that protein that play different roles. So class 2 has an alpha and a beta chain. So it's a heterodimer just like class 1, but for class 2, let's see, MHC class 2, I need a better pen, is going to have this alpha and beta. And I kind of drawn these with a little bit more of an open-ended structure because they combine to longer antigens. These combine to 13 to 18 amino acids in length. They have a tendency, and I think they've shown that these can even bind to whole proteins that later get chewed back. So they can bind to really long proteins. And then in the endosome, as they traffic through up to the cell surface, there are enzymes chewing back on those. So what you end up with is this 13 to 18, is that what you said? 13 to 18 long amino acid peptides. So these are going to have present longer peptides. More information, different shape. They're also going to have little anchor residues that dictate which peptides we're going to present. And again, different people present different ones, which is why if my organ were to go into one of you guys and you didn't have the same alleles, my cells, my own cells, my kidney cells or my liver cells, they're going to present different little peptides. And so because I'm presenting different peptides, when you're making your own T cells, you're seeing one set of peptides and you're not seeing the peptides I'm presenting. So that when your T cells go around and see my liver, they're going to see my liver with peptides, in this case, with peptides that are being presented that you have T-cell receptors that recognize because you never had negative selection eliminate them. And so you're going to eliminate my liver because you think it's dangerous. That's why a lot of times when you get a transplant and it's not a perfect match, even when it's a close match, a lot of times they'll give you immunosuppressive drugs to suppress that potential T-cell response against that organ. So there are a lot of transplant drugs that are specifically target T-cells to try and avoid that rejection. So again, MHC class 2, a little bit different, both the alpha and beta chain. So you have alpha and beta chains that form both ends of this groove. And this groove, even though the picture doesn't make it look like it, is a little bit more open-ended than your MHC class 1 and allows for longer amino acid lengths to the peptides that fit in there. And so here's kind of just a cartoon view of your class 1, your class 2, and their involvement, and they're all, for example, class 1 has this transmembrane segment. Both the alpha and the beta on class 2 have a transmembrane segment. That beta 2 does not. So class 1 and class 2 molecules have polymorphism in the peptide binding region. So this is all the diversity in these class 1 and class 2 molecules are all localized to that peptide binding bind region. So, again, class 1 molecules, you find that diversity in the alpha 1 and alpha 2, not the alpha 3 regions. And in class 2, you find that diversity in the alpha 1 and beta 1 regions. And as you can see, when you look at these, the alpha 1, so the alpha 1 and beta and alpha 2 domains are inside of that groove. And again, with the class 2, you have an alpha 1 and beta 1 domain that are inside, that make up that groove. And that's where the diversity is within the population, within our species. So the nature of the peptide, this is, again, class 1, this is just a little synopsis. It's closed at both ends. This is, class 2 is open at both ends, so class 2 can actually bind to things that are larger than 18 amino acids, but they usually get chewed back before they're presented on the cell surface. There are anchor residues on both that dictate what peptides, so it kind of gives you a little bit of a decision on what peptides are being presented, but not specificity like you see in T-cell receptors or B-cell receptors. And nature of bound peptides, both ends interact with the MHC groove, but the middle arches up and away from the MHC, so that's class 1. Whereas class 2, it's held constant at a constant elevation. So just looking at the kind of the structure, these present, these peptides kind of sit in the groove as opposed to get kind of stuck up in the, if you think about it, kind of if you're limiting what's in here, you can see that those are going to stick out, whereas these guys kind of just lay in here represented. So there's several hundred different allelic variants in humans. There's up to 6 different class 1, 12 class 2 molecules expressed per person. So everybody has, so this is why it's really hard to match somebody, because you can have up to 6 class 1, different class 1 alleles, you can have up to 12 class 2 alleles, and we have to match, and a perfect match would be a match for every single one of those. Even to siblings, that's going to vary because whether you're a match. Identical twins would be the same. So it's always good to have a spare around, right? As long as you don't both have a genetic predisposition for whatever it is you need an organ for.
So how about a mother to son they still have pigment?
Yep. So, a given MHC molecule combines numerous different peptides. So these don't have specificity like an antibody or an antigen. And some peptides combine to several different MHC molecules. So we kind of loose and free with what they're presenting. So if you look at the structure, so how they figured this out is they eluded peptides from MHC. So they isolated MHC, they ran gels, they eluded peptides from these MHC molecules, and they looked at them, and they saw lots of different peptides, but they saw that in general they had commonalities at certain loci in those antigens, and the same for class 2. So you have certain or different MHC. So each MHC, so this is one allele, and this is a different allele. They have different anchor residues, but in general, it doesn't matter what's in the middle. It's what is at these anchor residues, whether they will bind to that MHC molecule. So that's, yeah.
How do they match people? How do they do it? They do genetic analysis? Or?
Yeah, usually it is genetic analysis. The
Sequence?
fastest and the easiest way to genotype somebody, or what they call it haplotyping. They're looking at your haplotype is what HLA moieties are. The different alleles are called HLA. So they HLA type you. And most of the times, HLA typing is they'll get a little blood, and then they will get DNA from that, and they're running that. Basically, it's a they're running PCR, and every different
Vaccines.
well, yeah, every different well has different primers, and those primers are gonna be specific for different alleles. So they're gonna recognize, so that diversity, they are gonna, a lot of those primers end with whatever, different,
So you're just
codon.
adding everything else other than specific prime for
Yeah.
this well. Then when you rate it, to see if you have a band of whatever, you just use the fluorescence dye, then you can see, the fluorescence or not.
So it's a process. I've done it before. And they've kind of automated it now, so it's a lot quicker. But yeah, you would find out what alleles, and it's kind of cool. If you ever have
The
if
data already.
you're ever in a lab that does it, do yourself get your HLA type so that you know what
I
you
don't-
are.
So,
It's always fun. But if you're doing research, you don't want to find out what your HLA type is, because if you happen to be whatever you're studying in the lab, that turns you into the guinea pig, and you have to give lots of blood. It's better not to know. So class 1 and class 2 molecules, they have polymorphism in that peptide binding region, and that allows us to see what different alleles people are, because there is that difference in the region. They present peptides, again, class 1 presents to CD8 T cells. These are, again, cytotoxic T cells, and these guys, so MHC class 1 is on every cell in the body, every nucleate cell, which means that they aren't expressed on red blood cells, but all the other cells. Which is probably why things like malaria are so nasty, because we know our red blood cells aren't able to alert the immune system that they're infected with something. Peptides derived from endogenous, again, this is generality, but the majority of peptides presented via MHC class 2 are going to be endogenous. Some amino acids anchor the peptides into the groove, and other amino acids are available to interact with T cell receptors.
So these anchor molecules, are they conserved? Do they recommend this is certain?
The anchor molecules, that's where the variety is. That's where they aren't conserved. Well, they're conserved among alleles, and you would pass on certain alleles to your children, and your children would get the other half of their alleles from your husband, and then based on and they are conserved within alleles, but they vary. That's where your diversity comes from.
How many class 1 molecules do you have in person?
6, and then how many was it for 12 for the class 2? So here we kind of see. So even within, here's where that comes in. So when you look at, did I skip it? Yeah, class 2. So class 2 is the same deal as class 1, usually derived from, although antigens are usually, I'm glad that they put that in there, usually derived from exogenous. They put that in there because of our paper. Usually derived from exogenous extracellular processed antigen. They present antigen peptides to CD4 helper T cells, and they exhibit, they also have amino acid residue anchors that lock them into the groove, and they can oftentimes extend from the groove to interact with T cell receptors. So genes, looking at genes, MHC locus. So when we say MHC locus, these are specific kind of immune locuses that have 3 major classes of molecules: MHC class 1 or class 1 MHC genes, class 2 MHC genes, and class 3, which are going to be like your complement. And those have nothing to do with what we're talking about here, so you can kind of ignore class 3. They're not going to be involved in this antigen processing presentation. It is interesting that they are these complement proteins and inflammatory proteins do localize genetically speaking in these regions. not gonna make you, if you're gonna do research, it's good to memorize this. But for most people going into the clinic or going into labs where you're working with humans, it's more important to really know what genes are involved in humans. Again, if you are in research or going to be in research in the future, I would recommend kind of focusing in, memorizing these. But for humans, we have class 2, and we have gene DP, DQ, and DR. So this is oftentimes when we're talking about what alleles people have, you have HLA-DP, and then you may have DP 01 or 06, so you have different alleles of DP, different alleles, so you're expressing a DP, a DQ, and a DR, and you're expressing these from your mother and your father, and you have multiple alleles of certain of these, and then each DP is an alpha and a beta, DQ you have an alpha and a beta, and DR you have an alpha and a beta, so the unique alpha and beta for DP, DQ, and DR. For class 1, you have HLA-B, HLA-C, and HLA-A genes. So A, B, and C, that's easy for class 1. For class 2, it's DP, DQ, and DR. And where are those? So when we're looking at them on the chromosome, the basic layout of these MHC genes for humans is, and it's shown here. And so we have DP beta, DP alpha genes. We have some other accessory molecules that are involved in antigen processing and presentation, and we'll talk about that. So these are all associated. They don't play a role in, these aren't part of the MHC presentation molecule, but these are involved in processing antigens and loading them or editing what gets loaded on here, and we'll talk about those. You have your DQs, and then DRs, and then you have your class 1 molecules as well. Here just shows the exon and intron arrangement of class 1 and class 2 genes. Reflects their domain structure. Again, you don't have to memorize these. It's interesting to kind of see. And then here's showing the different allelic forms of MHC genes are inherited and linked to groups called haplotypes. So again, important to know what a haplotype is. A haplotype is going to be what HLA genes, what alleles do you express. And so if you are ever in a situation where you're talking about bone marrow transplants or any type of transplant situation, they're going to talk about haplotypes. Haplotyping is going to be, and then what MHC molecules does this person have versus another person. Each individual inherits one haplotype from each parent. Inbreeding of mice. So again, we've made mice really easy. So we've inbred them. So when we say that they certain haplotypes of mice, they're all going to be K. So you can have a mouse that is, that has all one allele. So we bred these mice to not have a lot of genetic diversity, so that we can study just one MHC molecule in them.
There's hundreds of types of haplotypes.
Yeah. So lots of diversity there. Unfortunately and unfortunately. So, I mean, we didn't evolve to do transplants. If we had, we might have, I don't know, done some other mechanism. But we evolved to not like foreign things, and so we developed this huge diversity. The problem comes in that when you're trying to do a transplant, there's huge diversity in what antigens you're presenting are different than what antigens I'm presenting, and now that's a problem. So both maternal and paternal MHC genes are expressed in offspring. This gives the best chance for an organism to have some capability of presenting all possible antigen peptides it encounters. This can also make transplantation somewhat difficult, as humans are heterozygous at each There's actually interesting studies that MHC molecules, and I don't know if it talks about this in the book, but are actually also involved in mate selection. So, and pheromones, and they've actually done studies that show that there's some genetic predisposition to choose a mate that has more varied alleles than you have, and that kind of drives more diversity within basically your haplotype. And so having a broader range haplotype has potentially better rates of survival if you have a pathogen or something like that. So this can make transplantation somewhat difficult, as I suggested. Non-matching MHC patterns will result in rejection. Some of these characteristics were originally defined in inbred mouse strains. So they did studies in mice where, so here's you have B mice, K of K mice, and they would cross them. So now you would have a heterozygous mouse that would have both, and then they would do skin transplants between them to see that if you did transplanted skin graft from a bee mouse, then it would not be rejected in a bee mouse, but in a K mouse it would. But in the, if you did that, so if you took a graft from one of these B of K mice, and you put them on either the B or the K, they were both rejected because these guys are presenting different antigens than both of them. And then, you know, you could do the same here. So that's kind of what helped us to give us this understanding of haplotypes and what the effect was in transplantation. So again, you know, here you can see that these B of K mice are going to have different, they're going to have, let's say, they're going to present some of this orange antigen and some of this green antigen. And so if you were to give that skin to another mouse, if they are recognizing orange antigen as foreign, they're going to reject that. And then if you put it into the yellow mice, they're going to see this is foreign and reject it. So kind of how to work.
So it's just like blood typing and A and B? Kind of
It's, yes, so blood typing, this is why you can get blood from people, because blood doesn't have MHC class 1 on it, but they do have these sugar antigens on them that will be recognized as foreign. And so blood typing is very direct, whether your immune system sees those guys or not, because it's a direct thing, a sugar on the surface of the red blood cell. Luckily, red blood cells, I guess, don't have MHC molecules, so you don't have that further level of rejection or not. Also, red blood cells only circulate for a certain amount of time anyway, so usually when you're getting blood, it's because you just need it until you start making your own red blood cells again. But so it's, yeah, it's similar. It's just it's a level that's a lot more.
But just in this simple diagram.
Yeah. So class 1, class 2 molecules exhibit diversity at individual and the species level. Obviously, mice are going to present different antigens than humans. They have different completely MHC molecules. Not only are you going to recognize different antigens if you have mouse tissue, but you're going to recognize the MHC molecule as foreign itself. And so,
Do we need to know all of the mice stuff? It doesn't need to know nothing, right?
yeah, unless you're in research, but I'm not going to ask questions on mouse. I might use the mouse as an example in a question, but you're not going to be reliant on memorizing what's class 1, class 2 on a mouse. It'll be more of a mechanism type of question. Like if you have a mouse that has this allele and a different mouse that has this allele, just like the picture I showed you, what skin will be rejected in which case or something like that. You had a question? Where's that diversity? Here we're looking at the variability, and if you look at the gene and the residue number, where that variability lies tends to be within. So here the red sections are showing you where these different parts of the protein lie, and that variability is always within this region.
The red region?
Yeah. Yeah, so differences tend to be clustered at the amino acid locations within the groove site. Like I said, this makes sense because that's where they decide which peptide to bind, and that's where they edit what they're going to present. Class 1 presents intracellular antigens and includes cell proteins, provides a way of checking that the cells are self and generally healthy. Can also be used to show which cells have been infected with viruses. Class 2 is more restricted, generally found on cells involved, so these are APCs, help to direct the response against threats, things that shouldn't be in our system. Class 1 expression is found throughout the body. Class 2 is only on antigen-presenting cells. Again, things that I've already mentioned. MHC expression can change with changing condition, genetic, regulatory, so promoters that drive up transcription during times of infection, so you actually can increase MHC production based on inflammation, based on different promoters can upregulate more MHC. Viruses like to shut down MHC class 1 expression because it targets the cells that they are in. Some cytokines expressed during infection drive up or down MHC expression. And MHC class 2 alleles and antigen presentation, different capabilities to present antigens may dictate the overall strength of the response. But again, whether you mount a response against a class a CD8 cell that counts in response or not may depend on whether a CD4 cell also got a signal. CD8 and CD4 T cells can only recognize peptides presented by MHC class 1 and 2 respectively, and so how they figured this out was isolating basically you can the experiment that kind of showed that is they had two mice, either strain 2 or 13 or a 213 heterozygous mouse, and they injected antigen into one of them, waited 7 days, so for this waited for T cells to develop, and then they isolated a lymph node, which is the source of a lot of T cells, and then they had a column. You can have adherent columns or plates, and what happens is a lot of your antigen-presenting cells will adhere to, they're very sticky, and they'll stick to the plate or they'll stick to the beads in the column, and this is an easy way to isolate either, in this case, they isolated antigen-presenting cells. In this way, they washed off all the cells that didn't adhere, which were all the T cells, and here, they collected the T cells, and so they were able to isolate either antigen-presenting cells or T cells. Here, they added antigen to the antigen-presenting cells, allowed them to process that antigen, and then they were able to mix these two together, and then they measured T cell proliferation, and they were able to show that if you had antigen prime T cells from strain 2 and you mix those with strain 13, that they did not recognize antigen being presented by the MHC molecules of strain 13, but they did recognize antigen being presented by the allele of strain 2, or of both, because this had both the 2 allele and the 13 allele, so MHC, this had at least an MHC molecule that presented that antigen. So this is kind of the experiment that they showed that different antigens are being presented by different MHC molecules, and that T-cell receptors are going to be specific for those. again, when T cells go through positive selection, they're recognizing your own MHC molecule. So if they don't recognize your own MHC in that positive selection, they're deleted. Okay, so this is just saying that all antigens can't be recognized by the T cells like they are antibodies. Okay, so T cells only recognize antigen. So if I have an antigen that the T cell recognizes and is floating around in the blood, your T cell is not going to bind to that. It will only bind to that antigen if it's bound to an MHC molecule. So how do these MHC molecules process antigen, and how do we make class 1 present endogenous antigens and class 2 present exogenous antigens? And so here in this figure, antigens are processed a little bit differently. Class 1 presentation requires cytosolic or endogenous processing, and class 2 presentation requires exogenous processing. So here, if you look in this picture, so here's our class 1. Antigens are being processed. Old proteins are being targeted, ubiquitinated, targeted to the proteasome. They're shunted through. The proteasome chops them up into little pieces. They come in through a molecule called TAP. They get loaded onto MHC class 1, and then class 1 binds to these little peptides, and some peptides it will bind to, some it won't, based on the allele. And then that MHC class 1 molecule will now traffic through the Golgi apparatus up to the cell surface and present antigen. Whereas class 2 doesn't bind to antigen in the ER to a major extent, and somehow is able to traffic, and then will only bind to antigen that has been brought in through the endosome and through these kind of acidic environments, in these endosome environments, you're going to get chewed up. So kind of like lysosomes will chew up proteins. So these are so antigen presenting cells are phagocytosing or pinocytosing or endocytosing exogenous antigen, chewing it up and then mixing it up with those MHC molecules and presenting that on the cell surface. So so class 1. So we're going to start with class 1. This is class 1.
Time.
Huh? Are we out of time?